Let's start today. Hi, everyone. We start today with Christophe Pouzat, which will talk about working memory and metastability in synaptic neurons model.
Thank you, Julien. So I want first to thank the organizers for inviting me and for making this meeting happen. It's, as we all know, it's not that easy right now. I tend to speak too fast, so if you can't understand what I'm saying, don't hesitate to make noise so that I can try to repeat. What I'm going to talk about today is joint work with Morgan André, who's now at UNICAMP. And so what am I going to tell you? First, I will present you some neurophysiological data about working memory and a specific paradigm used in neurophysiology to study working memory. That's called delayed response. And that go through the observation of persistent activity in neurons. Once you would be familiar with that, I'll define a very simple model that we designed with Morgan, the idea being to have something that is simple to implement, efficient on a computer, and that we can manipulate analytically. I will then show you empirical results from simulations of this model. And I will keep going with a mean field analysis, very simple, in fact, that allows us to get most of the properties of the network in the metastable state. And I will conclude with perspectives. Okay, so what's working memory? What's a delayed response? So working memory is what you use when you're lost in a city, your cell phone is not working anymore, you have to ask someone in the street for your way, and this person tells you, okay, you turn first left, second right, and so on. So for a while, you have to remember that you have to turn first left, second right. But once you've reached your goal, you're not interested anymore in these informations, and you can forget them. It's the same when you do a calculation in your head. You want to store transient results. You will use working memory. I mean, most of what you do intellectually requires working memory. The question is how we can study this phenomenon in a neurobiological setting. And the first person who did that is Joachim Foerster in the early 70s. And in his first paper, big paper on the subject, he says, okay, a delayed response trial typically consists of a presentation of one of two possible visual cues, an ensuing period of enforced delay, and at the end of it, a choice of motor response in accord with a cue. The temporal separation between cue and response is the principal element making the delayed response procedure a test of an operationally defined short-term memory function. What does it mean? That's illustrated here. You have on the left the monkey. The monkey can see two boxes when the blind is up. You can see that there's some holes in the head of the monkey so that we can bring down electrodes to record the activity of some neurons. At the beginning of the experiment, the monkey is shown the content of the two boxes. One of the boxes contains most of the time something that the monkey likes, and the other one is empty. Once the monkey has seen the content of the boxes, the blind is lowered, and for a few seconds or even a minute, the experimentalists wait, raise again the blind, and then the monkey is allowed to open one of the boxes. And if he chooses the right box, it's allowed to take the food contained in the box. So that's the paradigm that would be used in most of the experiments I would describe. What you see on the right are recordings made during this task. So the cue epoch is represented with a bar, and the vertical bars you see here are what we call raster plots. That's the time at which a given neuron, a single neuron, emits action potentials. And most of the time I will refer to action potentials as spikes. So my spikes have nothing to do with the famous spike protein right now, okay? These are just the action potentials of the neurons. So what you see are repetition of several trials. The arrow at the right tells you when the blind is taken out and the monkey can access to the boxes. You have C, the R here means that the reward is on the right side, the L means it's on the left side. So you see that the neuron is not active before the cue is presented. And after the cue presentation, the neuron becomes active. At the time the blind is taken away, the activity stops. And you see that the activity doesn't depend on what side the reward is on. It can be right or left, the activity is there. So it seems to be linked to the fact that the monkey must remember that for a while that something interesting is on the left or on the right side. These are more examples from another neuron, and this time you have some variations because sometimes there's no bait, so nothing interesting in any of the boxes. So in fact, the monkey doesn't need to remember that there's something because there's nothing to remember. And then you see that the neuron is not active. So again, that enforces the idea that this activity is really linked to the fact that the monkey must remember something. Now that we have more sophisticated experimental tools, very often what people do is that they train a monkey to look at a computer screen, as we all do right now, and they are trying to fixate the central point. And during the cue period, one spot is shown. Then it's tough. During the delay period, the monkey must keep its eye centered. And when the signal comes, usually a sound, it must make an eye movement towards where the light was during the cue period. The recordings are made in this part of the brain that's called the prefrontal cortex. And you see on the lower left here, something that you see better here. So that's always the same neuron that you see active. And the raster plots are located in relation to where the cue is located. So you see that this neuron remembers that cues are at the bottom, the south, okay? And if the cue is anywhere else, there's no modulation of the activity. But again, you see that activity appears when the cue is presented, it lasts during the delay period, and when the signal comes, the activity goes away. If you change the delay, so these are two examples, again, the same neuron with a short delay and a long delay, you can see that this sustained activity persists as long as the delay lasts. What happens when the monkey does a mistake? You have an illustration here where the preferred direction of this neuron is left. And in this trial, the activity disappears spontaneously. And then when the signal comes, the monkey makes a saccade to the upper right corner. So it makes a mistake. Okay. Other example, you have here a neuron whose preferred direction is the upper left corner. But for this trial, the activity is not turned on, and the monkey, when the signal comes, moves its eyes to the south, so to the bottom. So you see that you have a link between the activity of the neuron and the fact that the monkey does the task right or not. And particularly interesting for me is that in some occasions you see this activity disappearing spontaneously. More sophisticated paradigms yet. Here, the monkey touches a stick, a plastic stick, and the stick is vibrating at a given frequency. So that's the cue. And after a delay, it starts vibrating again, and the monkey must decide if it's the same frequency of vibration or not. Here you have a neuron that responds specifically to these stimulations. And what you see is that faster the vibration rate during the cue, the higher the rate of a sustained activity. So it's a bit like if the the neuron was not only representing the fact that something has to be remembered, but it remembers the quantity of this thing. So the faster the vibration, the higher the rate of the discharge. So that's called parametric working memory. So as you can guess, and these experiments have been going on for a long time, started in the early 70s. And many different labs have reproduced this kind of experiments. So that's something that is really reliable. You should never take what neurophysiologists say at face value. I mean, until you have 3 or 4 labs that have repeated the same thing, you have to be careful. But in that case, many labs have reproduced these findings. So modelers have started working on the subject very early on, early 90s. At the beginning, they used a lot of models based on differential equations representing the activity of small groups of neurons. And you have a first review by Xiaojing Wang in the early 2000s, making giving you a panorama of what was done at that time. Now, what about the virtual neurons that we saw in these experiments? There's a very interesting paper from 2006 from these guys, where they did experiments using slices of ferret prefrontal cortex using multiple patch clamp recording. So they put several electrodes and they can record several neurons at once. And what they find, subnetworks of specific pyramidal cells. So pyramidal cells are the excitatory cells that you have in the cortex. They represent roughly 80% of the cells in the cortex. And so you have subnetwork of pyramidal cells that are hyper reciprocally connected and whose synapse display a pronounced augmentation. What does it mean? That's illustrated here. You have 2 neurons, the black one and the green one, that are recorded at the same time. When they stimulate the black neuron, so that's the tiny thing you see here are action potentials evoked by the experimentalist in the black neuron. That's the response at the top in the green neuron. You see that if you increase the rate at which you stimulate the black neuron, the response in the green neuron is enhanced. It becomes larger and larger, but you have a facilitation effect. So the second amplitude is larger than the first, even if you subtract the delayed part of the first. And that's very peculiar. Most of the pyramidal cells in the cortex, when you do this kind of experiments, you have a decreasing response, not an increasing one. So you have this very uncommon group of cells. They are highly connected one with the other, and they have this facilitating synapse. So that led people think that this sustained activity could be explained by, so you have a quiescent activity, then the cue comes in, and the cue boosts the activity of a group of neurons. This group of neurons is highly interconnected, and because their synapses display facilitation, when you start exciting them, you make their synapses more efficient. And when the cue goes away, because you have this short-term facilitation, the activity of the network, of the subnetwork, is sustained. Okay, and that was again studied with differential equation-based models, so deterministic models. And you have a recent review from 2014 here. You can find a handout corresponding to this talk where you have all the references. It will be put by Lauren on the website, or you can go on my website and find it if you want. So what do we want to know? I mean, I've spent a lot of time recording from actual neurons, and for me, neurons are stochastic units. And they are stochastic units for 2 main reasons. The first one is that the channels that are responsible for the action potentials and everything, electrically wise, are stochastic units themselves. So what you have here is a famous paper by Sikroff and Ayer from the early 80s. Each trace here represents the activity of a single sodium channel. When the trace is at the top, the channel is closed. When it's at the bottom, it's open. and you repeat the same simulation again and again. And you see that from time to time, you have a blip corresponding to the opening of a channel. If you average 300 such traces, you get this. Okay, and what is responsible for the upstroke of the the upward part of the action potential is the opening of these guys. So you expect that if you don't have too many of them, you will have some fluctuations. Now, when neurons are talking one together with chemical synapses, look at the upper right part here. Neurotransmitter is released in packets that correspond to physical vesicles. And when you repeat the same simulation at a low rate, what you get as a postsynaptic current is shown here. So sometimes there's nothing. Sometimes you have a tiny response, sometimes you have a large response. And at the top of these large fluctuations, you have smaller fluctuations that come from the opening and closing of ion channels. So for me, neurons are stochastic units, and therefore with Morgan, we wanted to build a model taking stochastic units as a building block. So what is our model? We will take a system with n identical neurons. So again, you can think of the pyramidal cells of the paper I showed you before. Each neuron is synaptically connected to all the others, because in this subgroup in the prefrontal cortex, they find very high connectivity among the subnetwork, the members of the subnetwork. To each neuron, we associate a membrane potential ui of t that takes value in n. So I take value in n because I want to use integers in my code. It requires less memory and it runs faster. It's just a technical detail. Then we put a threshold. If the membrane potential is below the threshold, the neuron cannot spike. If the membrane potential is above the threshold, the neuron spikes at rate beta. When the neuron spikes, its membrane potential is reset to 0. And that's the only way the membrane potential can decrease. So that's a gross approximation compared to actual neurons. To each neuron, I associate a facilitation variable that we write f of I that takes value in 0 and 1. So if fi of t is 1, then that means that if a spike occurs at time t for neuron I, then it will increase the membrane potential of all the other neurons by 1. If fi of t is 0, when the spike occurs, there's no increase in the postsynaptic potential of the neurons. The facilitation state of a given neuron is set to 1 immediately after a spike that's been emitted by this neuron. And the facilitation is lost at rate lambda. Okay. So in fact, we are making a very simple model of the subnetwork described by Wang et al. In 2006. So what does it give? You have here a network simulated with 50 neurons, beta equals 10, lambda 6.7, and threshold of 5. In fact, you don't need to have beta and lambda separately. If you decide that beta gives you the 1 over beta is the time unit, what counts is the ratio lambda over beta. We'll see that soon. So you see everything relevant to make to predict the behavior of the network. The traces, so each trace is the membrane potential of 1 neuron. You have 50 such traces. It goes from 0 to 5, because if the membrane potential is above 5, you don't have to keep track of a precise value. You know that the neuron can spike. You see that the traces alternate between blue and orange. When it's blue, that means the synapse of the neuron is facilitated. When it's orange, the synapse of the neuron is not facilitated. If I zoom for 0.05 time units, so let's look at what goes on here. You see, you have 1 neuron that is in the upper state, so it can spike, and it's blue, so its synapse is facilitated. And suddenly, the membrane potential goes down. So that means the neuron emits a spike. And you see that all the other neurons that can increase their membrane potential do it. Okay. Now you have another neuron that is also in the upper state. So it can spike, but its synapse is not activated, is not facilitated anymore. So when it spikes, the other guys don't change their membrane potential. Okay, but when it spikes, it turns from orange to blue, so its synapse became facilitated. Okay, so that's the basic dynamics of this network. So how does it behave empirically? So it's a pure Markov process. It's very easy to simulate. You just have to draw exponential random variables to do it. My C code outputs things that looks like this. In a text file, you first have parameters used for the simulations, including the seeds of a random number generator. And then you have the time at which spikes are emitted. You have a neuron of origin. You have the number of neurons that are at or above threshold at that time, the number of active synapses at that time, and if the synapse was of that neuron was active or not. So if I represent now what you saw before, but I just put a dot when you have a downward trajectory of a membrane potential, okay, when you have a spike. So each row corresponds to one neuron, and the dot is blue if the synapse of the neuron that spikes was facilitated at the time of the spike, and it's orange otherwise. So that's, you see a total of 14 time units here, and the activity disappears spontaneously around times 13. What you have, so that's between the scale bar goes from times 12 to times 14. And this region is enlarged here. So at the beginning, you have roughly half of the points that are blue, half of the points that are orange. If you look at the details of what goes on at the time when the activity goes away, you see that at the end, all the dots are orange. So at the end, when the neuron spike, their synapses are not facilitated anymore. So that means they won't bring the membrane potential of the other guys up. And that's how the activity will go away. And that's the same network with the same initial state, but with a different seed to run the dynamics. And that runs for 50 time units without any apparent large fluctuations. And again, you have this mix of blue and orange dots. So the representation I will often use is the counting process associated to the whole network. So that's a function that jumps by 1 unit every time one of the neurons of the network spikes. Okay, and the red trace corresponds to this simulation. The black one corresponds to that simulation. So it dies after 13 time units. Okay, so the key point is that before the first simulation dies, it behaves as far as overall activity of a network exactly as the other simulation that will last for the time of the simulation. Okay, and I guess you see where I want to come. This spontaneous disappearance of the activity reminds me of a mistake of a monkey I showed you before. So we could potentially try to explain this sustained activity with this kind of mechanism driven by short-term facilitation. What happens if we change lambda, keeping everything else the same? That's illustrated here on single simulations. You have lambda equals 1, 2, 3, 4, 5, here 7, 8, and 9. So the point is, again, you have a very regular activity of the network. The smaller lambda, the higher the activity of the network. And when lambda becomes too large, the activity tends to disappear soon. So the fact that we can modulate the rate of the network by changing lambda suggests one mechanism for the parametric short-term memory I showed you before, when the monkey had this plastic stick vibrating at its finger. Okay, I'm not saying it's that, but it could be an explanation. I mean, a mechanism by which it would be represented. So now in my abstract, I said that our network exhibits a strong form of metastability. The reason I say it's strong form is because if you make a search on Google or on your favorite web search engine of brain and metastability, you will have thousands of references coming. I mean, every time people see vaguely constant in the brain, they talk about metastability. Here we mean, when we speak about metastability, we mean that it's memoryless. So if you are in the metastable state, it stays the same, but it will stay the same for an exponentially distributed time. So what you see here are the results of 1,000 replicates with different values of lambda. That's lambda equals 6, 2 with lambda equals 6.7, and 1 with lambda equals 7, 50 neurons, so exactly the same conditions as before. And so I run 1,000 different replicates. They all start with the same initial state, except these two guys. And what you see is the probability to be still active a given time after the beginning of the simulation. Okay, so that's the survival function. And be careful, it's a log scale. So you have straight lines on a log scale. That means you have an exponentially distributed survival time, which is so the key argument we have empirically to claim that our model exhibits a strong form of metastability. If you keep increasing lambda, so that's lambda equals 7, that's the last one you saw here. Okay, so you see the scale goes from 0 to 500 here. The scale goes from 0 to 5 here. And so lambda equals 7, 8, 9, 10, and so on. So you see that when lambda becomes too large, you lose this straightness, and you have this concave shape. So as if you were losing the capability of the network to exhibit metastability. So how does, on average, the survival time behave as a function of lambda? That's illustrated here. Again, log scale on the ordinate. These are 95% confidence intervals. That's computed as from the likelihood ratio. And there's some censoring for the smallest values of lambda. Okay, so my likelihood takes care of this censoring of some of the simulations. So you see that for lambda small enough, you have more or less a straight line, so as if it were you had an exponential behavior. And when lambda becomes larger and larger, then it decreases less fast. How does the survival time behave with the network size? That's illustrated here. Again, a log scale. Now we have something that looks like a straight line, so compatible with an exponential growth of the survival time with network size. So that's for the empirical results about this model. Now, what we do what physicists do and what I'm capable of doing. So I cannot prove, or we cannot prove yet, that the metastable state exists, but the simulations show that it looks to be there. So I will do as if it was there. We use the intrinsic symmetry of the model, so all the neurons are equivalent, and the goal is to get some network properties in the metastable state, like the network varying rate, the number of neurons in each state, and so on, depending on the 4 parameters, which are in fact 3, because what counts is the ratio lambda over beta. So how do we do? So we have our membrane potentials for each neuron that live in N, but in fact, everything that matters is U below theta or not. So in fact, I just have to keep track of theta plus 1 different states, 0, 1, theta minus 1, and then equal or above theta. So I will write Ni with I equals 0, 1, theta minus 1, the number of neurons in the state I, and N theta, the number of neurons whose memory potential is theta or above. Then at every time I have necessarily the fact that if I sum over these ni's, the sum equals n. And under the assumption of quasi-stationarity, the expectation of the ni should be almost constant in the metastable state. So I should have quantities mu0, mu1, mu theta constant, such that the expected value of each of the n's is roughly given by these mu's. Okay. If I manage to compute mu theta, so the mean number of neurons at or above threshold, then I have the network rate. Okay, the network rate nu n is just mu theta times beta, since all the neurons fire independently when they are above threshold. Now the critical quantities comes. So in our model, when neuron J spikes at times S, its facilitation variable just after S is 1. The question is, if J spikes again at time S plus tau, do we still have the facilitation variable equal to 1? But by our model definition and the quasi-stationary assumption, we have that the expected value of f at S plus tau given tau equals exponential minus lambda tau. Okay, it doesn't matter when the spike occurred. All that matters is the elapsed time between two successive spikes. Then I introduce my second key quantity, which is mu e, the expected value of exponential minus lambda tau. The expectation is taken with respect to random variable t, whose realization is tau. So loosely speaking, mu e is the mean probability that a synapse is still facilitated when the neuron spikes. Okay. If I manage to compute mu e, then I can define the effective spike rate, which is just the network spiking rate, mu theta beta, times mu e. So that the rate at which the network generates spikes that will make the other neurons' membrane potential go up. The stationarity means that the rate at which the neuron leave the membrane potential states 0, 1, and so on must equal the rate at which the neurons enter that state. Okay, we have conservation of the total number of neurons, so we have a cycle from state 0, 1, 2, 3, and so on. And we say that we are stationary, so we should have roughly at every time the same number of guys in each of the, and a constant number of guys in each of the states. So for the states from I to theta minus 1, so let's take I equals 1. We have here the rate of active spikes that will push all the neurons in state 1 to state 2. And we want that to equal the rate, the number of neurons from state 0 that will climb into state 1. So that's again the rate of effective spikes times the number of guys in state 0. So, and these guys must be equal. So that means we must have an equal population in each of the states below threshold. Now, if we take care of the two extreme states, the number of neurons, or the rate at which neurons leave state 0 is given here. And the rate at which neurons enter state 0 is given on the right side. So that's the rate at which your network spikes. So that means that we have mu theta beta in both sides. So mu e must equal 1 over mu 0. And we must have the conservation at every time. So we get that mu theta equals the network size minus the threshold divided by mu e. So we see that if mu e increases, then mu theta increases also. And if mu theta increases, nu n increases. Okay, so an increase in mu e will lead to an increase in the network spiking rate. And we can also obtain an expression for the effective spiking rate, which is given here. So beta mu e n minus theta. So now what we want to get is an implicit solution an implicit equation satisfied by mu e. So in the metastable state, a neuron leaves a membrane potential state below threshold at rate beta mu en minus theta. And that neuron must go through a succession of theta states to reach threshold. The distribution of the time to reach threshold is as for an Erlang distribution with parameter theta and beta mu en minus theta. So the mean value of this Erlang distribution is this quantity. Once the threshold is reached, the rate at which the neuron spikes is beta. So the interval between two successive spikes of a neuron is approximately, so the time it takes to reach threshold and then an exponential random variable with parameter beta. Okay, so I'm replacing the Erlang random variable by its mean. We want to get mu e, which is the expected value of that guy. So we have mu e approximately equals to that quantity, okay? And I obtain this approximate value for mu e. So you see that if you divide everything by beta, and all that matters is the ratio lambda over beta. And you see that if you fix theta as a fraction of n, then n doesn't matter because it will go away from here and here. So that's an implicit equation that we must solve for mu e. We can do better than that, taking the full Erlang random variable instead of the mean. We need to make some numerical integration that can be done. But I will show you that doesn't change much. So if we look again at this approximate value for mu e, we see that the right-hand side is a decreasing function of lambda. So if lambda is too large, the equation could have no solution. And if we have no solution, then we have no metastable state. Okay. So what does it look like? These are drawings of the right-hand side here for lambda equals 6, 6.7, 7, 8, 9, and so on. The dotted blue lines in these two cases are what I get with the exact solution, not replacing the Erlang distribution by its mean. You see that it's very close. So we have the key features that we observed. If lambda increases, so we go down, we see that mu e becomes smaller. If mu e becomes smaller, the rate of the network goes down. And if lambda is too large, then there's no intercept anymore, and there's no solution for mu e, so there's no metastable state anymore. So we think that, and remember before, my exponential behavior over several time disappeared slightly before this value. So I'm close to what I actually observed in the simulations. So if I compare what this method gives me for mu e and then the other network parameters with a network of size 500, beta equals 10, lambda equals 6, and so on, with one among many simulations, you see that my predictions with the mean field are very close to what I observe. Okay. So what do I conclude? So first, we have to start working seriously because we must prove the existence of this metastable state. Then from a physiological viewpoint, since that's my real field of expertise, the model suggests that we could change the rate of a network by playing with the value of lambda. And to my knowledge, there's not been any studies of how experimentally the rate at which facilitation is lost carried out. And then if our hypothesis that this delayed response is due to this self-entrained mechanism driven by this short-term facilitation, then that means we should lose memory, so start making mistakes more and more frequently in an exponential way as the delay between the queue and the signal increased. And that could be tested with psychological experiments on humans. And with that, I thank you all for listening, and I'm ready to answer or try to answer your questions.
Okay, thanks a lot, Christophe, for this nice talk. Do we have any question from the audience?
I have a question.
Okay, Quentin, go on.
I was a bit surprised in your model, but maybe I have not understood correctly. It seems that you do not scale the interactions between the neurons with the number of neurons, which means that as n goes to infinity.
Okay, sorry. I forgot to point out. Thank you for the question. I raise theta. My theta is always a fraction of n. So all the simulations you saw, theta equals n over 10. So for this, for technical reasons, I want to keep an integer value for my memory potential.
I see.
So as you rightly point out, most of the time when people increase the size of a network, they decrease the synaptic weight. So instead of decreasing the synaptic weight, I increase the threshold.
But my point is, when you do that, the problem is, I think, is that it's not simple to write a limit equation for the system with an infinite number of neurons, because it's not clear that there should be an equation for the mean field for the mean field limit.
Yeah. No, I agree. I mean, it's not bound to work.
I believe it would be simpler to study the metastable properties of the networks if you could write a mean field equations and then study the invariant measure of this mean field. You see what I mean?
Yeah. Okay.
I don't know, maybe.
Yeah, no. I mean, I'm just starting. I mean, I'm not. I was not raised in that field. I'm learning the tools. So, I mean, any suggestion is welcome at that stage.
But anyway, it was really interesting. Thank you.
Thanks.
Okay, we have a question in the chat. I can read it if you want. So it seems that the implicit equation for mu e admits two solutions.
Yeah. Okay. Yes.
But you choose only the larger one.
Okay. So I try to observe the smallest one without success so far. I think it's unstable. So I mean, as soon as you go a bit away from it, most of the time you die, or perhaps you go and you move towards the other one. But yeah, that's a good point. The implicit equation indeed has two solutions, and I couldn't observe the lowest one.
Yeah, if I understood correctly, in your simulations, as soon as you go away from the first, let's say, metastable point,
everything dies. Yes.
Yeah. Okay.
Yes. So yeah, I haven't tried super hard, but I've tried to observe the lowest one. I couldn't so far.
Okay, maybe I have one question.
Mm-hmm.
Okay. So you were saying that as you think the parametric working memory thing should be linked with the beta parameter in your model, if I understood correctly. That is the exponential decay rate of the facilitating state.
Yes, the lambda. Yeah.
Lambda. Sorry. Yeah, and yeah. So you were talking about lambda, and I was wondering if it was more linked with beta. I would have said, because if I understood correctly, the beta is more linked with the stimulus, no? Or
except that during the delayed response, you have no stimulus left. So the stimulus or the Q, if you want, brings the subnetwork, so to speak, high.
Okay. Yeah.
And when the Q goes away, so the stimulation goes away, and you have a self-entrained activity that is there. And then when the signal comes that the animal must make a choice, you have a new input to the network that brings down the activity. So that's so you could, since what counts is the ratio lambda over beta, you could also change beta in principle. My bet is that it's if a mechanism like that is used, it's more likely to be lambda than beta.
Okay.
Because, so beta is the rate at which you spike above threshold. Yeah. Now you could also get that. That's right. I mean, but let's say it would be easier to study if it were at the lambda level, because that will imply so what is responsible for the short-term synaptic facilitation is residual calcium in the presynaptic terminal. So when the spike arrives, it provokes the entry of calcium into the presynaptic terminal, and that's this increased calcium concentration that provokes the fusion of vesicles, so the transmitter release. And this calcium takes time to be pumped out of a presynaptic terminal. And the proteins that are the physical pumps can be modified by phosphate groups that could change their rate. So there's potential ways, known ways by which you could change lambda. That's what I mean.
Okay, so let's say the physiological or the biology behind this facilitating state is quite known, and this change in the lambda rate also.
So we understand pretty well what goes into lambda. So it's an interplay between, in the presynaptic terminal, you will find proteins that are specialized into binding reversibly to calcium. So cells are, every cells, not only neurons, are designed to keep a steady low level of calcium. And to that end, they are equipped with proteins that are buffering the calcium concentration. And so calcium binds in a very reversible way to these proteins. And then you have also in the plasma membrane other proteins that are pumps that will take, so to speak, calcium ions in and put them out. And you have the same kind of pumps on the mitochondria. So they will take calcium out of the lumen and bring them inside the mitochondria. And the kinetics of calcium is an interplay between these proteins that are buffering calcium and the pumps.
Okay.
But I should say, to be complete, and that's in the handout, that's not the whole story of this facilitation, because these synapses involve on the postsynaptic side NMDA, and you have mechanisms with the NMDA channels that will also lead to a transient increase. So in that specific case, you have probably two mechanisms at play, one on the presynaptic side and one on the postsynaptic side. That doesn't change much with the very idealized modeling we did, but at the physiological level, you have two different mechanisms at play.
Okay. Okay. Are there any other questions? Okay. Maybe just for the culture, let's say, do you know if there is a real, let's say, statistic study of what you shown at the beginning, the fact that the neuron frequency is really linked with the stimulus frequency, and also because the first, I was wondering, yeah, the first picture that you showed, the statistics did not seem to be different. No, the first one. So sorry, yeah, I was confusing those two. Yeah, here is the statistics of the spike trend different when the response is right or left?
you don't have enough data to tell. So the data, you have to realize that these are very tough experiments to make, because so you have to train the monkey to perform the task. That takes 3 to 6 months, rather 6 months. And then the monkey gets bored. Okay, so you can't make many tries at every session you make. And you don't record the same neurons from one session to the next.
Okay, yeah.
So as statisticians or modelers, we are always asking experimentalists to get more data.
Yeah.
But for them, it's really tough.
Okay.
Plus, I mean,
but for the direction, for the 6th direction, I don't know, 8th direction.
Yeah.
It was more clear.
Yeah.
Okay.
So that's from 1989. Sorry, the first experiments were from 73. So that's so now we are using much more electrodes at once. So every experiment leads to more data. In the 70s, they couldn't do that. So you find now much better data than you found 50 years ago, definitely. Yes.
Of course. Okay. Thanks a lot. I don't know if we have no other question. Maybe we have a 5-minute break before Bjorn talk. Okay, so let's meet again at